Renowned chemist Marie Curie once observed, "If I see anything vital around me, it is just that spirit of adventure that is indestructible and akin to curiosity." All of you here tonight are our community of curiosity. It is your spirit of adventure and inquiry that binds us all together and has made the Watson Lectures an enduring program at Caltech for more than a century. So welcome to the 102nd season of the Watson Lectures at Caltech. I hope you guys got a chance to hang out outside, listen to some music, and talk to the students. There's students from the Clemens lab and the Diversity in Chemistry Initiative. And you know what? I gotta say, it's good to be back. I'm Crystal Dilworth. I'm your host. I'm a science communicator and a Caltech alum. I did my PhD here in the Division of Chemistry and Chemical Engineering. And since then, I built a career highlighting the humanity behind the scientific endeavor. And the Watson Lectures is one of my favorite ways to do that because we can pull back the curtain to see the people that are actually doing the science. The Watson Lectures began in 1922 by Caltech professor Ernest C. Watson. It was in response to a conversation that he had with a high school teacher who was having a hard time keeping up with all of the scientific innovations that were coming out that he wanted to share with his students. And so Dr. Watson's solution was to just invite the entire public to Caltech. And he put on awesome and highly popular physics demonstrations. There were talks about the innovations and the ideas of the age. And he really made what was going on within the Caltech ivory tower accessible to the wider community. And because of those origins, I want to take a moment to kind of recognize any high school students that we have in the audience, especially those from Roscoe Tech, who, as members of our junior Watson program, had the opportunity to come to tour the lab of Professor Bil Clemons and, of course, hear about the opportunities available to them in science and technology, and at Caltech especially. For tonight's adventure, we're going to be hearing about how, in the future, we might be combating pandemics with viruses. But before we do that, you're going to hear a little bit about tonight's speaker, Professor Bil Clemons, as a person. You'll hear an introduction from his friend and division chair, Dr. Sarah Reisman, and then you'll watch a video about his life and work. After his talk is when you get to ask your questions. So you'll see a QR code on the screen. Take a picture of that with your phone. It'll send you to our online platform. You can submit your questions for Dr. Clemons in that way. Now, please remember, questions are short and they contain a question mark. You know, I just thought it might be worth mentioning. If you're old school, raise your hand and we'll have ushers that will see you and make sure that you get a comment card to write your question on so that we get all of those questions answered. And then afterwards, to end the evening, tea and coffee on the patio. There'll be an opportunity again to maybe ask some follow-up questions for Dr. Clemons and, of course, interact again with this wonderful community that we've built here. All right, I think that's all of the business. So if you can, take out your phones, silence your watches, silence your phones, all of your tech that beeps, note your nearest exit, and allow me to welcome to the stage Caltech's chair of the Division of Chemistry and Chemical Engineering, Dr. Sarah Reisman.
Hi, good evening and welcome. It's my real pleasure and privilege to introduce tonight's Watson lecturer, my friend and colleague in the Division of Chemistry and Chemical Engineering, Professor Bil Clemons. As you'll hear more about in a minute, Bil grew up as part of a military family, living in several cities throughout his childhood before finishing high school in Virginia and attending college at Virginia Tech. Bill earned his BS from Virginia Tech and then his PhD from the University of Utah, working in the group of Professor Venki Ramakrishnan. During this time, he spent 2 years as a visiting scientist at the MRC Laboratory of Molecular Biology in Cambridge, England, and this also happens to be where he met his wife, Jana. As a graduate student, Bill played a key role in solving the structure of the small ribosomal subunit, work that was later recognized when Professor Ramakrishnan shared the 2009 Nobel Prize in Chemistry for studies of the structure and function of the ribosome. Following his graduate studies, Bill was a postdoctoral researcher at Harvard Medical School, working with Professors Tom Rappaport and Steve Harrison. And then he began his independent career here at Caltech in 2006 as an assistant professor of biochemistry. He was promoted with tenure in 2013, and then he is currently the Arthur and Marian Hanisch Memorial Professor of Biochemistry. Bill directs a research group in the area of structural biology using X-ray crystallography and electron microscopy and a variety of other biochemical tools to study problems related to protein transport across membranes and the post-translational modification of proteins. This work has important applications in human health, for example, in the development of new antibiotics. In addition to his research, Bill is an advocate for diversity, equity, and inclusion in science and in higher education. He has served on the President's Diversity Council at Caltech. He helped to found the Black at Caltech group here on campus, and he is currently the Science Program Officer for Diversity, Equity, and Inclusion in Science at the Chan Zuckerberg Initiative. He was awarded the Shirley Malcolm Prize for Excellence in Mentorship, and if you've spent any time with Bill, you know that he is passionate about the human side of science. He's a strong advocate for our students, and he has worked tirelessly to make Caltech a better place for all. Bill's research has been recognized with several honors over his career, beyond the Nobel Prize to his thesis advisor, including an NIH Pioneer Award, his recognition as a Searle Scholar, his election to the National Academy of Sciences. Bill's also a fellow of the American Society for Biochemistry and Molecular Biology and a fellow of the American Academy of Arts and Sciences. Bill lives in South Pasadena with his wife Jana and his sons Kuba and Ozzy and his dog Mila. I feel very fortunate that I call Bill and his family my close friends. And in fact, during my interview visit to Caltech, when I was deciding if I would also join the faculty, Bill and Jana attended my recruitment dinner, and in fact, it was their first dinner away from their oldest son, their newborn son, Kuba, and so I felt very fortunate to have them join me. And they were instrumental in helping my husband, Andy, and I settle in the Caltech community. Bill loves the Boston Celtics and Arsenal FC, and he enjoys playing board games, including the game Pandemic. So it's perhaps apt that his lecture tonight is named Combating Future Pandemics with Viruses. Please join me in welcoming Professor Bil Clemons. We'll first see a short video.
Many members of my family are musicians, and I was intrinsically a scientist from as far back as I can remember, to the point that I wasn't allowed to play with my siblings' toys until they were done with them, because I had a tendency to want to take them apart and see how they worked. I had this one story where my brother and I had gathered up all these frogs, and there's probably 100 of them in a box, and we thought, wouldn't it be cool to see them swim? And so we went to the bathtub in the house and dumped in all these frogs. We watched them swim for a little while, and we ran off. And about 20 minutes later, we hear this scream from the house, which was my mom discovering a bathtub full of frogs. I'm Bil Clemons, the Hanisch Memorial Professor of Biochemistry at Caltech, and my lab studies membrane protein biogenesis and ways to target bacteria. My dad was in the military, and so I was born in Virginia, Quantico, Virginia, where my dad was in school there as a Marine. And so we moved back and forth across the country, in California, North Carolina, South Carolina, Hawaii. My dad's brother, he was a sax player, and he met this guy, Bruce Springsteen. Our family grew up with my Uncle Clarence being the sax player for the E Street Band. He was just my uncle, but he's also this person who was on stage, right? And so to watch your uncle and now my brother get flashed up on the camera and hear the whole stadium roar, it's pretty good. But what you appreciate from that is excellence, excellence all the time. I mean, to get to that level, you had to do that against everything else. And that translates to some extent in science as well. I think in the vast majority of it's just hard work. One of the unexpected gifts that my parents gave me, they didn't see at the time, was where a salesman sold them a bunch of encyclopedias. I'd pick up a volume and just sort of read through it out of curiosity. And it was just, this idea that there's just so much I can learn and so much you can know. That was always driving force for me. When I was looking at majors in college, do I want to do biology or chemistry, or, biochemistry, I can do both. My undergraduate research advisor kind of kicked me in the butt one day and said, hey, you're good at science, you should pursue this. And I was lucky that he did. And so when I got to Utah, my first encounter with Venki, we call him Venki, even though he's Sir Venki Ramakrishnan, Nobel laureate now. But sat in his office and he said, understanding the structure of the whole ribosome, that's the sort of, that's the goal. Like if we can figure out how to do that, which, any person who was seasoned in science at that point in time would have said, you're crazy, I'm not going to do that, that'll never work. I was young, very naive, so it sounded great to me. What a great problem to work on. Our team basically going from starting from nothing to having an atomic resolution structure by 2000 really was, it was crazy. One of the greatest things about Caltech is our students. They're curious and hardworking people who really want to solve life's mysteries. And my lab's no exception to that. I've got a team of really amazing, thoughtful, clever individuals who are making discoveries every day. We've worked anywhere from archaea to bacteria to mammalian systems. We're now projects in plants. Almost all of those projects could only start because I had colleagues who were willing to just let us come and use their equipment, show up in their labs, sit in their offices and talk science. And I think there's only a few places in the world that are as special as that, where the doors aren't closed, where you can talk to a mathematician, you can talk to a geologist, you can have these kinds of conversations on a daily basis. That enables you then to ask the kinds of questions that other people can't ask. The reality is, as you pursue problems, if you're not deeply invested in the answer and you're not driven by that discovery, that potential discovery, you'll never get there.
Okay. All right. So welcome. Thank you all for coming. It's really a pleasure to be able to take the stage here in front of you all, and I'm grateful that you were curious enough to come and hear about the science that my lab's been working on. Of course, I stand before you here, and I'm going to tell you some science stories, and hopefully you'll find them interesting. But I do want to acknowledge, of course, that I stand here before you. Obviously I put in a lot of work, but I didn't do it alone. And of course, I do want to recognize first and foremost my family, my wife, who has been an important support structure for me through my entire career. But I also wanted to acknowledge the fact that I'm lucky enough to be here at Caltech, where I have a team around me of amazing people, and that team includes my colleague Doug Reese and all of our lab mates that have joined us on this research journey. And of course, Mila, that's Mila there in the bottom. She's showed up a lot in the video. So this is really an awesome team, and it's really incredible, and it's really fortunate for me to have the opportunity to be able to come into work every day and talk science with these people, learn from these people, see what they've discovered, and be able to actually communicate that to the world. But most importantly, I want to highlight three people who really are the drivers of the stories I'm going to tell you today. And so we're going to really, when we get to it, we're going to talk about kind of a couple of simple science stories. The first story is really led by two people, Nadi Riera, who was a former postdoc in the group, and Karen Orda, who recently got her PhD and is about to be a former grad student. She's leaving in, I think, 2 weeks, sadly. They always grow up. And then, of course, the final part of the story is going to be work from another grad student in the lab, Evelyn Lee. And so we'll get there in a minute. So I do want to highlight the fact that I actually wear two hats. So in addition to being a professor here at Caltech, which is my passion and my joy, I've also taken a role at the Chan Zuckerberg Initiative, where my title is officially the Program Officer for Diversity in Science. And throughout my career as an academic, it's been important to me to think about ways that we can ensure a diverse science workforce or a diverse science environment. We want to create a space where everybody has access. And that's important because everyone can do science, and diverse science is better science. And so CZI gave me this opportunity really to do this on a national scale. And I won't talk more about it, but I'm happy to answer questions if people are curious. I just want to point out, we get to do cool things like this. So this is what we call a fireside chat, where I'm hanging out with Rick Kittles. So Rick Kittles is the vice president for research at Morehouse Medical College in Atlanta. Rick's a really amazing guy, but in this fireside chat, he was telling us about the importance of historically black medical colleges to the success of the United States, and it was really a great story. So the science story really talks about phage, and it really centers on this idea about using phages as mechanisms for both discovery and also as tools. And I think one of the great stories of phage and sort of our modern era really starts with this couple here, Steph Strathy and Tom Patterson. Their story is actually a beautiful one. They've told it much better than I can tell it here. But they basically were on vacation, and Tom developed a systemic infection from a bacteria called Acetobacter baumannii. You don't need to know Acetobacter baumannii except to say that it's a pretty friendly bacteria until it develops resistance to all known drugs. And that's what happened in this case. So he had an infection of a bacteria that was resistant to every drug that we had that we thought could treat bacteria. And he was dying, he was in a coma, and the doctors told him that there was no solution. Fortunately, his wife, Steph, was an epidemiologist. She knew medicine, she knew lots of doctors, she knew science, and she just turned to the literature and just started pouring through papers until she discovered phage. And she brought together a bunch of doctors and phage biologists, and effectively he was the first person to be cured of a systemic infection of phage in the United States because of her efforts. And so they detail the story in this book, The Perfect Predator, and I would encourage you, if you're interested in the story, to dig into that. It's really a fun, well-written book. So, none of us remember what life was like before penicillin, right? But the reality is that death was around the corner everywhere you looked. And, we don't think about these things as sort of scary things anymore, right? But pneumonia, cholera, the Black Death, diphtheria, plague was everywhere. People were dying in huge numbers from bacterial infections. And in fact, you can talk about lots of different numbers in terms of birth rates and complications. But 10% of the time that you got an infected cut would result in death, or would result in loss of a limb or death. And you can think of these things like scratching a mosquito bite, right? Can you imagine back in the day that you're sleeping and you get bitten in the night by a mosquito and you scratch it absentmindedly, it gets infected, and the next thing you know, you're having your whole arm chopped off, right? I mean, this was reality for people prior to the discovery of drugs. But of course, that changed about 100 years ago. And the first commercial drug that came on were a class of drugs called sulfa drugs. Prontosil was the first commercial drug that came out. Some people actually maybe remember taking sulfa drugs. They're still used today, not very common. But this was the first example of this back in 1932. It was the first one that was commercially available. You're probably more familiar with a drug that looks like this, a penicillin. And so, of course, Alexander Fleming discovered penicillin accidentally when he found a mold growing on a Petri dish. But that mold was secreting this molecule here called penicillin F. Now, we're going to talk about molecules and structures here a lot today, and there's lots of different ways to represent them. One way to represent them is like we see here, where it's this flat surface of the structure here. But these actually are three-dimensional objects, and so I want you to experience them as three-dimensional objects. And so now's the time, for those of you who haven't put your glasses on, you don't need to have your glasses on the whole time, but now is a good time to put your glasses on, because hopefully what you'll see is that this molecule is now rotated in 3 dimensions, right? And so these molecules have depth. They exist as 3-dimensional objects. And protein molecules, everything that we want to see and interact with and think about, has this 3-dimensionality to it. And so we need to be thinking about these things this way. Just as an FYI, as we go through the talk, if you see a video with a black background or you see an image with a black background, in only one case, which I'll point out, it'll be a stereo picture. Here's another molecule. This molecule is called amoxicillin. Right now, I forgot to point out, but penicillins are all derived by having this beta-lactam ring. So you can see there's that square and that pentameric ring. That's called the beta-lactam ring, and that's sort of an essential piece of penicillins and related molecules. Just a quick show of hands. How many people have either taken amoxicillin or given amoxicillin to somebody? Wow, impressive. People read the label. Good job. Yeah, everybody, right? In fact, I didn't encounter amoxicillin as a real thing until I became a parent, because amoxicillin is the major drug that we use to treat ear and throat infections in children, right? It's that pink stuff that you put in the fridge and you have to use a syringe, right? And so this is a really important central drug that we use now, and it was discovered in 1972. And it still works, okay? So you're gonna come away from this scared after this talk. The reality is we still have lots of good drugs, but the threats are rising. The reality is that, and so this is just a chart, a timeline actually showing antibiotic drugs that have been discovered, various classes on the left, and on the right is the years that clinical resistance was discovered for those drugs. And so if you follow on the left, what you'll see is that, sure, we keep discovering new classes of drugs, but more recently, every single drug we discover, we effectively at the same time find resistance. And that's never gonna stop. Bacteria are amazing. Evolution is powerful, right? We will always evolve to find solutions. The bacteria will always evolve to find solutions around drugs, even if we think we're clever. Evolution is more clever. And this even goes to the latest drug. So this drug here, tixobactin, is probably the most exciting antibacterial on the market right now. It was discovered in 2015. Its mechanism of action is a completely new mechanism of action, which is exciting. It's still in clinical trials right now, so it hasn't made it to the market. And I say this is a new drug, and that's exciting. The problem is that we know resistance is going to develop. And so the way that we avoid that is we don't actually allow you to take it unless you really, really, really need it. And so what that means then, if you're a company, you're a pharmaceutical company trying to develop drugs, you really don't want to give a drug that develop a drug no one will use, right? So there's no money in it, right? So this is a major problem. And it's growing, right? So, resistance to drugs is happening worldwide. It's being discovered worldwide. This drug, colistin, is currently our drug of last resort. So if you are resistant to everything else and you're in the clinic, they will give you an IV drip with colistin, and hopefully that will save you. But in reality, bacteria have already been discovered in our water supply last year, in our wastewater, that are resistant to colistin, right? So this is not just a faraway problem. This is a problem here today. And the estimates are, from the WHO, that by 2050, as many people will die from antimicrobial resistant bacteria as will die from cancer, right? You just keep that in mind. And so between now and 2050, if you take that number, an estimated 39 million people will die from these, what we call them, superbugs, right? That's 1 every 20 seconds, right? And so we have to solve this problem. What should we do? Right? Well, of course, we're going to develop new drugs, but that requires investment. That requires investments because the people who are going to do that are academics, right? Companies aren't going to do this, right? Small startups will try to do this, and there'll be investments from government into companies, and that's where the money really is going into developing new therapeutics. But the reality is that we have to support this work directly. The other thing is to find alternatives, right? And these alternatives I've already hinted at is, bacteriophage. And so this is a cartoon of a bacteriophage. It's a T4 phage. And if I was to say phage to you, and if you've heard of it before, you probably picture something that looks like this, right? This lunar lander molecule. Bacteriophages aren't that special in the sort of whole scheme of things because they're really just viruses, right? But they're viruses that have specialized to target bacteria. Now, we draw these models because we actually can see these things. And this is just an negative stain electron microscopy images where we're visualizing these T4 phages here. And even to the point that you can use a scanning electron microscope, you actually can see bacteria that's being targeted by these bacteriophage, right? Bacteriophage are just viruses. And what do viruses have to do? They have to find their host. They have to infect their host. They have to inject their genome into it some way. Once they've injected their genome, they have to take over that bacteria, force the bacteria to make copies of itself, assemble new particles, and then escape, right? And that's the life cycle of phage, the life cycle of all viruses, right? You have to get in, force to make copies of yourself, and then get out. The problem for viruses is they can't make copies by themselves. They need a host to be able to infect. For bacteria, the sort of interesting thing about bacteria is that they have to actually breach the cell wall to be able to do that, right? They have to escape the bacteria, have to break it open, effectively killing the bacteria, which is bad if you're a bacteria, but it's a good thing for us. All right, here's another opportunity to put those glasses back on. So what we have on the left here is actually a structure of this top of this bacteriophage here. So this is the phage, the head of the phage here. It is, it's where all the genetic information is stored, right? And give this a second. So this head, I hope you can see this in 3D. We put a lot of thought into making this work. This phage head is large, right? There's thousands of proteins that go into making this. And the reason why it's so large is because it has to contain a genome of 160,000 base pairs. And that's not big by our standards, right? We're in the billions for humans. But for a virus, that's a really big genome. And the reason it has to be so big is it has to do all that stuff, right? It's got to make all these particles, it'll make this structure, it's got to assemble them, it's got to take over genomes, it's got to escape. T4 is a cool phage, but the phage that we're going to get excited about is this phage here, phages-174. Now these are drawn approximately to scale. Phages-174, you can see, is much smaller than the T4 phage. And because it's smaller, it can only hold a small genome. And that small genome has to do all the same stuff that you do with the large phage. It has to find its host, get in, make copies of itself, and get back out again. All right, glasses off. So bacteriophages were discovered roughly 100 years ago by two people, one of those people being Felix de Herelle. Felix de Herelle is a fascinating character in science history. If you are bored one day and you want to get a good Wikipedia read, look up Felix de Herelle. He's an amazing guy. He was French-Canadian, but he did some of his seminal work in Paris, which is where he discovered bacteriophage. And the thing that was remarkable about Felix de Herelle is when he discovered bacteriophage, he immediately made the connection that bacteriophage killed bacteria. There's a virus that kills bacteria, and you can use it to cure a bacterial infection. And in fact, he was the first person to cure a patient using bacteriophage. So he made a cocktail of bacteriophage, gave it to a 12-year-old with cholera, and the next day the kid was over the disease. Right? Really remarkable. So this was exciting. He started a whole laboratory in Paris. They started collecting phages from everywhere they could find. Basically, where do you find phage? Wherever the bacteria lives, so sewers, right? They were collecting phages from as many different sewers as they could find. And they were preparing them, testing them in lots of different things. They saw this as a real opportunity for the development of therapeutics. And a sort of interesting thing about this lab that he established in Paris was there were a lot of Eastern Europeans who had become interested in this research. One of those being this guy, George Eliava. George Eliava had started a microbiology institute in Tbilisi in Georgia, and he'd come to visit and learn about phage, and they actually took de Herelle back with him to Georgia to establish a research facility there. And so there's the Eliava Institute for Phage Biology in Tbilisi. The problem was that institute was heavily supported by Stalin. It became the Soviet cure. And at about the same time, this Soviet cure was now in competition with pharma, because about the same time that they were getting these things to work, actually to start figuring out how to solve the problem of curing patients, penicillin was being marketed, right? Now you have this magic drug in the West. And so in the West, people basically said, we don't need phage anymore. So they completely stopped doing research on phage as a medical therapy. Whereas in the former Soviet states, it continued up until this day. And in fact, if you visit the former Soviet states, if you were to go into a pharmacy there, you can get things like this called peophage, which is just it's a cocktail of bacteriophage that if you have a stomach infection of some sort, they give this to you, you drink it, and it cures you of your stomach infection. So amazing. That's not true here. You can't do that here. Maybe one day. But this story from Steph Strathy really changed the game, right? So when Steph cured Tom of this disease using bacteriophage, it really opened the door to being able to do this research in the US for medicine. And they're actually actively, she founded an institute called IPATH in San Diego where they're actively curing patients using bacteriophage. And in fact, bacteriophage clinical trials, since their discovery around 2016 when this story happened, you can see that lots of different startups have started, clinical trials are en route, trying to use phages as a cure. It's really exciting. It's an exciting time to be in phage. All right, now we're going to change gears again. We have a lot of changing gears here, but now for science and Caltech. All right, well, the first thing you need to think about is bacteria. This is not stereo. I forgot that this one's in black, but it's not stereo. This is an E. coli, right? This is an artist rendition that comes for us from David Goodsell down at Scripps. He's an incredible scientist and artist, and he does these really beautiful renditions of bacteria and all of the different cellular life based on things that we know from structural biologists. If you zoom in here, this is an E. coli bacteria, and we don't need to know much about it. We're going to zoom into that square there up on the top, where we get a little bit more high-resolution image here. And so what do we see in this picture? Well, the first thing you can see is something that you probably would have guessed already, which is the genome, is here in red, right? That's where the genetic information is stored. Now, my PhD was spent trying to understand the structure of these purple things here called ribosomes. And so the genetic information from the DNA gets transferred into mRNA, and it gets processed by those ribosomes to make protein. And so you can see these ribosomes are reading along the mRNA. Another key part here is the cell wall. And this defines the outside of the cell. This is an E. coli. It's a double membrane bacteria. So it's got an inner membrane and an outer membrane. The membrane we're most interested in is the inner membrane, which is called the cytoplasmic membrane. And it basically is that sort of inner layer there. Now, the one thing you'll note is that inner membrane is actually studded full of proteins. Okay, and so there's proteins all over the place here. And those proteins are called membrane proteins. You know a little bit about membrane proteins, so we're going to do a little primer here. This video, as I'm showing you here, is actually a molecular dynamics simulation, so it's not actually real. We can't actually visualize proteins. It's not stereo either. This is the one I meant to mention is not a stereo one. I should have done it stereo, but this would have taken a long time to make. This one is, but this video is a molecular dynamics simulation where we took a membrane protein and we embedded it in a lipid bilayer, and then we ran it computationally using Newtonian physics to calculate what it would happen on sort of nanosecond time scale. That bilayer that we see there, where the cytoplasmic membrane is exactly where we're looking at. It's composed of these small molecules that we call lipids. These small molecules have long carbon chains that don't want to be in water. They want to be, and because of that, they end up getting forced next to each other to form these lipid bilayers where water is excluded from. And that creates this membrane that we call the lipid bilayer. And what's great about that is that's really impermeable to anything that's polar, except we have to get things across: food, information, nutrients, whatever. And so evolution has created these membrane proteins as ways to get that across. And so embedded in this in green there is a membrane protein. Now, we have lots of different ways of representing proteins, and the way that we're going to look at them for the most part today is going to look like this. So proteins are just polymers that fold up into three-dimensional objects. And we can represent those polymers as atoms, which is a lot of information. But here, we're actually representing those as these alpha helices, which actually were discovered here at Caltech by Linus Pauling. Those alpha helices were represented as tubes, okay? So those are structural elements. Those alpha helices cross this membrane, and because they cross the membrane, they also have to be hydrophobic as well. And so these membrane proteins are really special. They have to exist in this lipid bilayer. Okay, finally, the other thing you need to take away from this slide is this little layer in sort of, I don't know what that color is, but between the inner and the outer membrane, this layer we call the peptidoglycan layer. All right, the peptidoglycan layer is critical to bacteria because it basically acts as an armor that surrounds the entire E. coli and really acts as almost like a chainmail of armor that surrounds the bacteria. What constitutes peptidoglycan? Peptidoglycan is actually made from pretty simple parts. It's got two sugars. Again, you don't need to know the chemistry here, but you can see in the orange and blue here are two sugars, one called N-acetylglucosamine and the other called N-acetylmuramic acid. Those two sugars are going to assemble together in a polymer that's going to be cross-linked by this pentapeptide, so these 5 amino acids which are going to be attached to that muramic acid. All right, that's as much as you need to know about that, except to say that these are going to be assembled together into these large meshes that basically encircle the entire bacteria. And the bacteria need this. It defines the shape of the bacteria. If you destroy the peptidoglycan, you kill the bacteria. So how is it made? Okay, this is where we get complicated. This is probably the most complicated slide, and you don't need to take a whole lot away from this, except to say we know how bacteria make peptidoglycan. They do it in a series of steps that basically starts here on the bottom with an enzyme called murA that basically converts an N-acetylglucosamine into this muranic acid, and eventually you add these amino acids to it. This creates at the top there this soluble component that we call Park's nucleotide, and we'll talk about that more in a minute. Park's nucleotide, that diphosphate, those two red circles there, one of those red circles is going to transfer with the sugar and the peptide onto the phospholipid there, catalyzed by an enzyme called MRAY. MRAY is an important enzyme, so we'll talk more about that. But that generates this first component that we call lipid 1. A second sugar gets added onto it to get lipid 2, and now we're starting to see something that looks a lot like what we were just looking at in terms of our peptidoglycan, right? That's the basic unit. Now, the problem with this is, on all my figures, the cytoplasm's on the bottom, the outside of the periplasm, this sort of outside the cytoplasm, is on the top. And all of the action for where the peptidoglycan gets made is on the other side. And so you need a flipase, this enzyme murJ, that catalyzes taking that head group across that lipid bilayer. Once you're there, a series of enzymes build the peptidoglycan. Now, of course, the peptidoglycan is unique to bacteria, so it's great for drugs, right? Penicillin, the first drug that you're probably ever heard of, targets this pathway, right? And since then, lots of drugs have come online that target this pathway. And this is not exhaustive at all. There's hundreds of drugs that target this pathway, different classes of penicillins and so forth. Now, for us, this is really exciting, but we're most interested in the membrane parts here. And the membrane parts turn out there are natural products that have been discovered that target these enzymes as well, but there are no clinical drugs. And so we're really interested in understanding the mechanism of how these works and developing these into clinical drugs. And we've got team members like Yusra and Isabel and others who are working to understand these mechanisms of these drugs and actually leverage those for new discovery. But today, what we're interested in is that phage have to figure out how to target these as well. And we're really going to start by talking about this phage phi-X, which, as you can see here, targets MRAY. Okay, so now for Caltech. So this is Robert Sinsheimer. Robert Sinsheimer was at the University of Iowa. He was a physicist who got into biology. He got excited about phage and met Max Delbruck, who recruited him to come to Caltech. He joined the faculty here in 1957. He became chair of biology, I guess in 1967, I think is the correct date for that. He was chair here for 10 years of biology. But Sinsheimer was really the father of phi-X. And so phi-X was discovered actually in the 1940s, but no one was working on it from a sort of biophysical approach until Sinsheimer came along. And so he figured out how to purify it, he figured out how to work on it, and he started characterizing phi-X. And so when he came to Caltech, he established a lab here. And this is actually really cool. So I went to the Caltech archive and I said, do you have any pictures of Sinsheimer's group from around that time? And so this is Sinsheimer's group meeting here. And you probably don't recognize anybody except for Sinsheimer's there in the bow tie. But fortunately, a former alum who's here in the audience, Amy Lee, was able to actually identify all the people in this picture here. And so these were all colleagues of Amy around 1972, which is really cool. Amy actually has the honor of being Sinsheimer's last grad student. He left here to go into administration, and this is actually the two of them sharing a moment there. Okay, so phi-X174 is famous for many firsts in molecular biology. And we're going to go through these kind of quickly, but just appreciate the fact of how exciting this is. This phi-X174 was presumably in the first phage cocktail. In fact, if I was to go out into the audience, I could collect phi-X from this audience for sure. Some of us have phi-X inside of us. It was the first established single-stranded DNA that was discovered by Sinsheimer, partially while he was still in Iowa. When he got here, he basically established that it had circular DNA. He worked with Arthur Kornberg, who went on to win the Nobel Prize for RNA polymerase or DNA polymerase. But they basically demonstrated that you can make just from nucleotides DNA that could be functional, and so that was really exciting. The Sinsheimer lab went on to genetically identify all of the genes, and we'll see them in a moment. And then eventually, the first restriction map was discovered, and this is actually Amy's thesis project here. So, Professor Lee, it's really great meeting you today. So this is the first restriction map of any genome. Probably the thing it's most famous for is that it was the first genome that was sequenced. And so Fred Sanger worked on this, and actually Clyde Hutchison, who's an author here, was a grad student with Sinsheimer and went to England to work with Fred Sanger. And they sequenced this DNA, and it was actually the first, his second Nobel Prize for discovering how to sequence DNA. And so Sanger sequencing is a method we still use today, but the first thing that was sequenced was a phages 174. Hutchison actually went on to join the Craig Ventnor Institute, where he helped lead a team where actually the first synthetic genome, just purely from chemistry, was made, and that again was phages 174. And I highlight Professor Hutchison primarily for the fact that he was basically the person who discovered the gene that's going to be the thing that we're really excited about today. And this is how phages 174 kills its bacterial host. So phages looks like this. We have these structures of it. It's got a prophage, meaning that in the bacteria it looks like this. When it gets out of the bacteria, it releases that green protein and matures into the phage that can infect other bacteria. Its whole genome is here, reminding ourselves that it's a circular genome. It's about 5,400 base pairs, which is smaller, actually. Most grad students will tell you that's an extremely small genome. And we actually can know what all of these genes do, and in fact, we can actually associate many of these genes with actually parts of the structure. They play different roles. But the gene that we're most interested is in this gene here, which is lysis gene E. Now, lysis gene E is interesting because this is the only thing you need to kill the bacteria, is this gene here. We'll talk about this more. It's also the first example of a gene that was embedded within another gene. And what that means is the same stretch of DNA encodes for two different proteins, right? And it does it because the coding region is 3 nucleotides, and so if you shift by 1, you can make 2 different proteins. And so these proteins are actually genetically linked and evolutionarily linked as well. All right, so how do you kill a bacteria as a phage? Well, the phage we've been talking about, T4, does this spectacular explosion, right? It uses a series of proteins called endolysins, holins, and spanins to work in concert to time this release where it basically bursts the cells and all the phage get out. These smaller phages don't have the luxury of this extensive genome to encode all of these things. And so they have to do this with a single gene. So they do it much more subtly. In this case, they basically behave like a penicillin, where during cell division, you get a breakage of the cell wall and release of the cytoplasmic contents, and they do it with a single gene. And so this is a term that we use now that was actually coined by a colleague of ours, Rai Young, called single gene lysis, where we refer to these as sigils. Sigils are protein antibiotics that are sufficient for killing the bacteria when they're expressed. Phages is the first example of this, but there are many more examples of this, and this is a list of some of the more interesting ones. While phages is an example of a single-stranded DNA genome, all the rest of these are actually single-stranded RNA genomes, which actually has some interesting things to think about there. But the simple idea is you can take one of these genes, you can genetically put it into a bacteria. We measure bacteria growth based on something like this, where you've got test tubes, and you can see the different colorations here are just the fact that the density of bacteria has increased as the bacteria continue to grow. And we can measure that as a way called optical density. So we can start with this clear liquid, and we can watch our bacteria grow. And the difference between these 2 lines is in one line, we're not expressing a gene, and the other line, we're expressing this sigil gene, right? And what we see, remarkably, is after about 20 minutes, the cells start to die. Right? So this bacteria is making a protein, this peptide that kills itself. And we actually can visualize that here. So there, you might have missed it, right? We basically start with a bacteria that basically pops during cell division. You're going to watch on the bottom there, the little one on the bottom is going to pop there too. Struggling, trying to survive, and then it kills itself. And it kills itself because we forced it to make this gene. And so how do these things work? Well, we do know how one of them works because Rai Young's lab and others at Texas A&M had gone on to actually discover the mechanism for this. And so Q beta expresses this gene, which is on the surface of its protein shell, that actually binds the first enzyme in the pathway, murA. And it just acts like a sponge. It sucks up all of those enzymes. Those enzymes can't work. You can't make peptidoglycan. The next one that we're going to talk about is phaX. How does phaX work? Well, it expresses this protein called protein E, about 91 amino acids. It's got a single transmembrane domain and this long soluble tail. We got into this when I became friends with this guy, Tom Bernhardt. He's a professor at Harvard now, but we were postdocs when we first became friends. And he told me the story about his PhD where he was working with Rai Young at Texas A&M. And what Tom and Rai had discovered was the mechanism for how this worked, because they were able to show that they could identify mutations in MRAY, the central enzyme in peptidoglycan biogenesis, that would show resistance to protein E. And that implied that protein E had some effect on how MRAY worked. They also discovered this other protein, SLIDE. SLIDE is a chaperone. They knew it wasn't essential, so it wasn't clear what it was doing, but it was doing something. And so we'll see what it's doing in a minute. Okay, so we're going to go through and look at each of these parts here. Now glasses can come back on. All right, so this is a fun protein. This is MRAY, right? This protein, this structure was solved by the Lee lab at Duke. What you can see is MRAY is a homodimer, so there's two copies of MRAY that are back-to-back relative to each other. As we do the rotation, what you're going to see, we call them back-to-back because the active site's actually facing out. And so now, facing towards you is the active site. Now remember, the cytoplasm's on the bottom here, so that's where the active site lies, in sort of that cavity there in the back. This enzyme catalyzes this sort of complex reaction where it takes this C55P, which is this lipid substrate, which is going to diffuse in the membrane through the membrane into that pocket there. And then the other player is going to be the Parkes nucleotide, which is going to come up through the cytoplasm to dock into that. And this enzyme catalyzes a bond formation between this phosphate on the C55P and this phosphate that's attached to the sugar to create this high-energy diphosphate bond. The other player in this is SLIDE. So SLIDE is a chaperone. It's got two domains relative to each other. Now, when you see this motion here, this is because these are NMR structures. The NMR structures basically tell us that these proteins have two different domains that are well-ordered, but very flexible relative to each other. And so we know this chaperone is important. We know it binds proteins, so it's probably doing something along that. The final thing to point out is what phaX74 looks like. So, we can look at protein sequences. When we look at protein sequences, like as I said, we can isolate lots of different variants of phaX174. We can sequence all of them. We can look at their E gene, and we can align those together and ask, what are the important parts? And what you can see here is that the transmembrane domain, which really is this sort of blue region here, is almost completely conserved. So it's really essential to this process. And within it are these two residues here, which are actually prolines. So prolines are sort of interesting amino acids because they break secondary structure. And so they break helices, and so they're kind of unexpected to be in there. All right, so of course we wanted to solve the structure. And so my first postdoc who took this project on, Shigo Tanaka, she just asked that sort of simple question, can we actually purify these proteins together? So she expressed protein E, she expressed MRAY, and then formed a complex with SLIDE. So these formed a trimeric complex that we call the YES complex. Pretty much because it sounds cool, right? YES complex. And so the YES complex is all three proteins work together somehow. The genetics had suggested that, but now we had evidence biochemically. And so of course now we want to get structural evidence. So we turn to this tool that we call cryo-electron microscopy. Here we have our cryo-EM center down in the Beckman Institute, where my student Grace is loading a sample into this. So what we're going to do is we're going to purify those protein complexes. We're going to put them into ice, in a very thin layer of ice, and we're going to take pictures of those with electrons. Those pictures that we're going to take with this look like this, right? Now, I'm going to zoom in just so you can be clear. There is three-dimensional information here. We're going to be able to get to atomic resolution or near atomic resolution with images like this. But we do it by taking 12,000 of these movies. We identify all of these particles, so they get to the point like 12 million particles. Out of those particles, we can computationally average all of them to get a three-dimensional picture. And that three-dimensional picture looks like this. Now, I'm going to remind you, again, glasses back on, remind you that this protein exists in a lipid bilayer. But we've had to extract it from that lipid bilayer, and we do that using detergent, which are just small molecules that sort of simulate that hydrophobic environment. And so right now what you're looking at is the protein complex. All the proteins are there, except you're surrounded by the lipid bilayer. So that's not super interesting. We want to get, or the micelle. We want to get rid of the micelle and actually just focus on the protein. And what you can see here is that there are a bunch of different pieces now of the proteins that are engaged. We're going to look at these in more detail in a minute. But the one thing I want you to just appreciate is that this is the data that we take away, right? We get data that looks like this. We can zoom in on parts of it. So we're going to take that yellow helix there and zoom in on it. And what this actually is actually the molecules themselves, right? So we can take this image and we can build in our three-dimensional model of the molecules that we can see. We know what those polymers look like. We know what amino acids are there. And we can build this atomic resolution structure to get something that looks like this, right? And so now we have all of our proteins. So the first thing that you can see about the S complex is it's a trimer of dimers. There's 2 copies of MRAY, so there's 2 copies of everything else. There's 2 slide E's there on the bottom. There's 2 protein E's there in the middle. One of the things that's sort of interesting is that the slide E makes no context to MRAY. It makes no context to each other. And the reason is that slide E's and MRAYs have no interest in each other whatsoever. And so they shouldn't be coming together. But they do it because protein E brings them together. Right, so now we have this viral protein that has evolved to take over the bacteria and force it to make this complex. The effect of that is this transmembrane domain basically blocks. Remember where that active site is. You probably already guessed this. This yellow helix here that's running down through the front in this orientation is blocking that active site. It prevents the enzyme from being able to work. So here it's acting just like a drug by preventing this from being able to work. So what is slide E doing? I like to analogize this as a bow tie. It basically grabs onto that protein E, it locks the whole complex together, and it holds it in an architecture that basically is irreversible. Once this binds, you're stuck. Excuse me. So I had told you that those prolines were important, and I just want to point out here that if we look at this from this orientation, we're reviewing it from the side, those prolines are really where those breaks in the helices are. Right? So those prolines really are essential because they allow this transmembrane domain to be able to occupy this structure. We also can identify where all the mutations that Tom had discovered as a grad student lie. They all lie at the interface for where protein E binds to MRAY. All right, this is great. So now we can start filling in this picture here. We now understand the mechanism of a second sigil. So quickly in the last couple of minutes, I'm going to show you a few more of these. So now we're going to look at M. So sigil M expresses a sigil that's 37 amino acids long. And in this paper, they showed that this sigil targets MUR-J, that flipase. What was surprising is the paper came out more recently looking at this PP7 bacteriophage. And this PP7 bacteriophage has a 75 amino acids. The sequences are completely different from each other, but both of these sequences target MUR-J. So this phage had also evolved to target MUR-J. So what is MUR-J? Okay, so MUR-J is a flipase. It basically is going to take this head group and it's going to push it across the membrane. It does it through an alternating access mechanism. And so the head group is going to bind on the cytoplasmic side, and then as it converts to the periplasmic side, that head group is going to traverse the membrane in that fashion. And so how do these sigils work? And so this is the work of Evelyn. And so what Evelyn did was she solved the structure, just like we did for MRY. You could purify these complexes and solve them by cryo-EM. And what we could see is that for sigil M, it basically binds to MUR-J in this outward-facing conformation. It locks it in this orientation that it can't function anymore. This was remarkable because we actually have no inhibitors of these kinds of enzymes. This is a completely novel mechanism for killing a bacteria. This was unexpected. We didn't know what we were going to see, and here you can see it. It really has just wedged itself in there. So now the second question you can ask is, how does PP7 work? And this was really surprising. Almost exactly the same way. So this completely convergently evolved peptide binds to MUR-J and locks it in the same conformation. Now these peptides are very different sequences, right? So they can't bind in the same way. And in fact, where you saw with M, it comes back out. PP7, you can see it actually reaches into the middle there, and that C terminus goes down into the groove. And so now we can finish up so we're done with the glasses now. And so So now we can look at these sigil mechanisms, and we can basically identify now that we've filled this out. Now, you'll notice here I have 5 different phages here, and I've shown you 4. It's kind of interesting because MS2, we know what its sigil is. We have no idea what it does. We don't know how it works, which is super exciting, right? Because it means that there's exciting biology to be discovered if we figure out what that does. But it gets even better because these viruses are everywhere, right? These single-strand RNA viruses, and every single single-strand RNA virus we've discovered has a completely different mechanism for killing bacteria. And what that means is as we begin to understand these mechanisms, we're going to learn all new targets, just like we did with MUIR-J. But more importantly, we're going to learn ways to target those things. There's just new biology to be discovered. It's very exciting. And this is the kind of work that's being done by grad students in the lab like Rijan and Grace. All right, so I'm going to wrap up here and just say that phage biology is really central to our biomedical future. You know, phage therapy is obviously something that we're all going to be thinking about and talking about well into the future. But it's also it's a hotbed for discovery. And so the ability to learn from phages has taught us so much about ourselves, but it's also provided tools, and more importantly, it may provide therapies as we move forward. And we see this as a really tool for discovery. And so I will stop there and just say we had a lot of people who did this work, and hopefully we have some time for questions. Thank you.
Thank you so much, Bill. I'm glad that the 3D
Did it work?
worked.
Good.
Yeah. I felt like I got in a time machine.
Yeah.
I was looking, and everyone in the movie theater had their glasses on.
JAWS 3D all over again.
Yeah, absolutely. So, how many phages are out there?
You know, it depends on what you mean by how many phages. There's an infinite number of phages because phages constantly evolve. So there's nothing that you could point at and say, this is a phage, right? They're constantly changing. But if you do the numbers, I don't remember off the top of my head, you know, phages, like it's like 10 to the 30th phages on the planet, right? There's so many phages that you know, just from a mass perspective, they outpace us by a huge amount. You know, the oceans are full of phage. So if you take a mill of ocean water, 1 mill of ocean water has roughly 1 million phages in it, right? So you've drank lots of phages. Don't be afraid of phages. The other thing is that soil, feces, full of phages, about 1 million phage per gram in, you know, actively living soil. So, you know, phages are part of our lives. We just don't think about them.
You're going to need a lot of graduate students if you're going to pursue this research.
Yeah, I hope so.
So I have a bunch of questions here. One of them is, how do you know that the viruses that you use to fight pandemics won't rebel against us? Like, if they can infect bacteria, how long do we have to wait before they evolve to attack us?
Yeah, you know, it's interesting. I mean, you talk about viruses, and viruses are as old as life, right? I mean, viruses are everywhere. We're surrounded by viruses. You're covered in viruses. Viruses, just like bacteria, most of them are benign to us, right? There's only a few that go rogue and become problems. But that said, you know, I mean, evolution is really powerful, but for a bacteriophage to make the transition to a human virus is virtually impossible. You know, that could only happen, I mean, it just can't happen. You know, the things that they've recognized, you know, phages are specialists. You know, when I say they target bacteria, it's not like one phage targets all bacteria, right? Phages target a specific bacteria. And so for it to be able to target the bacteria of a different bacteria right next to that same bacteria is almost impossible. It takes a long time to figure out how to do that. So to make the leap to a eukaryotic cell would be huge. To make a leap to a human cell would be impossible.
It's interesting. That question was about going forward, and the next question is about going back. So the question is, can you potentially apply this method to fight any dangerous ancient microbes that could be released when permafrost melts? Because the planet is warming.
Well, yeah, I guess we're more worried about viruses than dangerous microbes. But no, the reality is phage. The beauty of phage is they've evolved to fight bacteria, right? And so, you know, using phage as a tool to combat bacteria, we're not redesigning anything. That's what they do. And so we're really just harnessing those as a tool. Now, of course, we can engineer them, we can improve them, we can use discoveries like this to make them better. But at the end of the day, they're doing what they were evolved to do.
Are bacterial pandemics inevitable?
No. I think, pandemics, we're not going to have the Black Death again, right? We're not worried about it at that level. The reality is what we're worried about is that, after a rain, you decide to go surfing anyway, and you get a cut, and you go to the hospital, and they're like, ooh, that infection looks really bad. And then they start trying to treat it, and it doesn't work, and it goes necrotic, and then you're going, what am I supposed to do? And, depending on where the cut is, it could get real bad real fast, right? So those are the kinds of things that we have to be worried about. You go to the hospital for one thing, and you pick up a hospital infection for something else. Bacteria don't spread like a flu virus or something like that, but they do spread enough that, lots of people are going to die. And, we all deal with bacterial infections often enough to know that because we have the tools now, we can fight them, but when we don't, it'll be different.
There's a question here about other potential mechanisms that phages can use besides targeting the peptidoglycan layer.
Yeah.
What else have we seen?
The problem, so it depends on the question. So these smaller phages are kind of different from these large T4 phages. These larger phages, they are impressive machines, right? They breach the walls, they take over the city, and then they force the city to stop doing everything it was doing before and just make phage particles. These smaller phages can't do all of that, right? So they breach the walls, but they just hang out and they convince, some part of the city to make small particles over time, and they'll just keep doing that. But in both cases, the phages need to get out, right? So you can kill the bacteria. Effectively, like a T4 phage effectively has already killed the bacteria before it's killed the bacteria, right? It's not, the bacteria can't make more bacteria, but it also can't get out until it breaches. And so breaching the cell wall is a critical component for all bacteriophage, because if you don't, you're now trapped in that chainmail, and you're just you're a bag now of phages that can't really do very much.
What role will AI play in developing these types of therapies?
AI, look, I'm excited about AI. I think AI has provided us lots of tools, and it's going to continue to do that. And I imagine a future where, at some point we'll know enough about bacteriophage. We'll know, those of us keep working on this biology, will know enough to understand both the mechanisms that phage use to attack and target bacteria, and also the mechanisms bacteria use to defend against bacteriophage. And if we've learned enough, we can begin to engineer bacteriophage, and then we can begin to, get large libraries of bacteriophage to the point that I can sequence your infection and then basically feed it into AI and say, what bacteriophage are going to be good against this infection? And it'll give you a list of, 50 phages, and you just push a button, and it gives you a syringe that you can move forward with.
That aspect of personalization that it already does.
Yeah, and that's the beauty of phage. Another thing about phage is they're specialists, right? They target a bacteria. So they're not going to do like an antibiotic, which just destroys everything. They're going to be very specialized in who they attack.
Shifting gears a little bit, I have two questions that I'm going to group together. The first one is, what is your favorite moment while working in CCE? And then the other one is, any advice on getting tenure?
All right, who asked that question? The reality is, I don't know about tenure, but I think being a scientist, the rule of thumb for me has always just been work on something you're excited about, right? If you're passionate about it, then everything else is easy, right? If you care about the questions and you're driven by the questions, then it's not hard to get tenure because you're working on something really interesting, right? And so I think that's the way that you should, if you want to be a successful scientist, you have to approach it that way. Wait, what was the first question? Now I've lost track.
What's your favorite moment?
My favorite moments, lots of exciting moments. I remember, watching live when Francis Arnold's Nobel Prize got announced. That was pretty cool. I remember the first time I saw the map for that Yes complex, and it was like, we've been working on that for about 10 years. We tell the story like it's simple, but it took a long time. And so we finally saw that and knew that we had it. It was so exciting. It was a really great day.
What is the most important aspect of mentoring students for you?
I think my life, I basically don't see myself as better than anyone else or worse than anyone else. I'm just a person like everybody else. And for students, I'm just farther along the journey. right? And so my goal isn't to somehow impress them with all the things I've learned in the 20 plus years I've had doing this. It's really to understand that this is where you're at, this is where you can be. But the way that you get here is you have to do the things to get there, right? And so when I mentor, and my relationship with students and hopefully with my colleagues and everyone is just, we're all at different stages of this road. And so mentoring is just helping people understand, they're on the right path, they're in the right space, and if they're not, how do they get back on that right path?
What can faculty do in terms of providing resources and guidance for students, especially when they are considering if they want to stay in academia or if they want to venture beyond the ivory tower?
Yeah, well, I mean, you tried to venture beyond, you're still here.
I was not successful.
Yeah, Crystal actually took my class, so I've known her since she was a first-year grad student. It was, I think the reality is everyone's got different roads, right? And I think, the sort of funny thing about getting a PhD is what we teach you is how to be a really great thinker, right? We teach you how to be a good scientist. But those skills are translatable to all kinds of other jobs, right? So it's not that, I think, we don't teach people to be academics. When I showed up, no one told me that, oh my God, this is how a budget works. Like, I had no clue, right? And so you had to learn all that on the fly. So I'd say, the thing that we do and the thing that we're really going for is really trying to train people to be good thinkers.
When can I buy Phage at CVS?
Well, there's probably a CVS in Georgia, maybe, the country, not the state. At some point, there are probably companies on the web you can buy Phage from right now. I would wager that. I haven't looked that up, but I'm sure that's true, right? And I think there are lots of people now who formed labs who are willing to help. The problem right now for phage as a therapy is that in the US, it's still considered a high-risk therapy. Therefore, you can only get it when you're desperate. And so by the time people get treated with phage therapy from a really bad infection, they're usually already in pretty bad shape. And so the hope is we can get over some of those hurdles so that people can get a little earlier than that. But my guess is because a lot of people are investing their time in it and hopefully more resources, we'll get there pretty fast. And my guess is many people in this audience will get phage therapy at some point.
Do you think phages are alive?
Well,
Robert?
Yeah. Do I think phages are alive? No. But I don't know what life is, right? I think the fact is that probably the sort of simplest definition of life is that you have to be able to make copies of yourself. And phages have to have a host to do it. So if there are only phages on the planet, there'd be no life on this planet, right? It's not true for bacteria, right? With bacteria, they would divide and evolve and get better. So I think that's the distinguishing point. I mean, you can talk about lots of mobile elements and sort of DNA and other things that jump from bacteria to bacteria, and those propagate themselves, right? And just like phages, they propagate themselves. So, if that's the definition of life, maybe, but I think that's a crude definition of life.
Well, thank you so much for being here and for sharing all of your amazing structures with us. And thank you all for being here. Remember to register for next month's Watson lecture as well. And please feel free to join us on the patio for tea, coffee, and perhaps more questions about what is life. Have a good evening.